Okay, there we go. All right, everybody, it's great to have you here for this webinar. Thanks for joining us. Good morning, good afternoon, good evening, good whatever time of day it is, wherever you are. I'm Gareth Dyke from the Charlesworth Group. This is our author education service here on the first slide. We are aiming to provide education for young researchers to help you achieve your potential and get your papers published, get your presentations up to scratch, and be as effective as possible at academic communication in English. So please, as we wait to get started on this webinar today, please check out our website, have a look at some of the other things, have a look at some of the other training materials that we have available that might help you in your career. So Tuesdays at the end of each month is the day that you can join us at the Charlesworth Group for our webinar series. And I'd like to ask you to please have a look at the other webinars that we've got available on this website link here. Why not spend the last Tuesday of each month with the Charlesworth Group learning something useful in just an hour that might help you to advance your career? You never know. We're about to get started with this webinar today. We're about to get started with this webinar today. As I said, my name's Gareth Dyke. I'm an academic author, published many papers myself, given lots of presentations, done lots of work as an academic, and for the last few years, I've been working now with the Charlesworth Group, helping people like you to gain the skills that you need to achieve your potential as a young researcher. And I'm extremely lucky, as we get started with this webinar today, to be joined by an associate professor from Harvard Medical School, Professor Tim Hall. Tim, are you there on the line?
I am, Gareth. It's a pleasure to be here today.
Excellent. So this is our presentation today, and we're going to talk for about 45, 50 minutes, something like that, about statistics and data presentation. It's going to be hopefully informative and interesting, and we try to keep it basic, because statistics and data presentation is something that if you don't do it well at a basic level, then there's no point getting complicated, in my opinion. So this is why we'd like you to stick around to the end of this webinar today. If you do stick around for the next 45, 50 minutes or so, we've got some free gifts to give out at the end as an incentive for you to stay with us through the whole of this recording. And also you'll have the opportunity to ask us questions. Ask Tim questions. He's the statistical expert after all at the end of this webinar. So if you have questions as we go through the material today, please put them into the chat box that should come up on the right hand side of your screen as you listen to our presentation. You're going to gain, I hope, a number of tricks and tips to effectively understand your data and learn a little bit about the basics of statistical analysis, as well as effective data presentation and how to sell the message of your academic work. So all of this, and you can get lots of generic content, lots of courses online, of course, like much of the content that we deliver is available elsewhere, but our goal at the Charlesworth Group is to help you achieve your potential, to more easily perform your statistical analyses, get your data presented in the best possible way, to get those papers and get those presentations into the best possible places, to really achieve your potential as a researcher. Very important at early stages in your career. So I don't know how to do statistics, but it doesn't matter because I didn't have data. This is crazy. Everybody has to do something analytical, usually as part of their academic career. And at least, even if your field is not highly numerical, as Tim's is, as my research field is to a little bit, you will nevertheless have to present data of some kind in your papers, in your conference presentations. And so we hope that this webinar will give you many of the tricks that you need to be able to achieve that. Tim, please feel free to interrupt me as we get started now with some of the core content for our webinar today.
Excellent. Gareth, a quick comment for you. I think it's amazing how many investigators got into science to do great science, but view the statistics and the data presentation as merely annoying. That
Totally.
they view they have to do it just to get their papers published, but they don't take the time to really get to know the methods or really do them well. So I'm excited to talk to everybody today to see if we can help the authors figure
That's
out
great.
ways to more effectively present their data.
And please keep in mind that you can contact Tim through the chat box on the side of the webinar today. You can ask him questions. He's an expert statistician. Him and a number of colleagues have developed an excellent software that we also use as part of our editing and pre-peer review services at the Charlesworth Group, a piece of statistical analytical review software called Stat Reviewer, which we'll talk about in a little bit as we go through the presentation, I think, won't we, Tim?
That's right. Excited to talk about that too.
Okay, so statistical analyses, an essential component of scientific research and a total must-have skill, really, for any researcher. But this is an example of a really key transferable skill that you will use, no matter whether or not you stay in academia for the rest of your career. These kinds of skills, presenting, analyzing your data, putting together figures that appropriately summarize the data that you've collected, are really important skills for any commercial, industrial, or even business working environment. That's the first half of the talk today. The second half on data presentation is an area in which we hope to help you effectively communicate your message clearly and effectively as you tell a story with your academic work. And as we talked about in previous webinars in this series, and you may have picked up from some of the other material that the Charlesworth Group has available online, selling the story of your academic research is the key thing to get your papers into high impact factor journals. If editors and reviewers aren't interested and captivated by the information in your scientific or academic article, they won't send it out for review. And that's often the stumbling block with many academic papers, getting them out through that initial editorial triage process. So bear in mind, statistical analysis and data presentation are what we're going to be talking about today. And this is Statistics 101, the science of collection, presentation, analysis, and reasonable data interpretation. We'll give you these slides at the end. You'll have this video, so I don't need to read out everything that's written on these slides, but the basic information that sets the scene for this kind of area. A rigorous scientific method for gaining insight into the data that you collect as part of your study. Doesn't matter what the data is, so long as it's collected and analyzed in a reasonable and reproducible fashion. This is the most important thing.
And Garrett, that's one of the neat things about statistics. As you said, it doesn't matter what you're studying. It doesn't matter if you're doing animal research, meta-analyses, randomized clinical trials, whatever you're doing, having a good core knowledge of statistical analysis and data presentation will help you get your research published and, more importantly, help your research have impact with your readers. And I think that's why it's so neat to be involved and to learn this stuff, because it is broadly applicable to whatever area you're in.
Absolutely. So we're talking about the science of statistics, which describes, doesn't it, a numeric set of data on the basis of its center, variability, and shape. And so we're looking at a categorical set of data that you may have collected as part of your research in terms of potentially things like frequencies or percentages or proportions that comprise the overall data set that you may have collected.
Right? So in most studies, researchers have to draw a sample from a larger population of things they could be looking at. So if it's animals, you do a small set of animals. If it's human beings, you create a sample from a large population that you could possibly study. You study that sample, and then you have to describe it, you have to analyze it. So I think that's what we're talking about now, is how do we analyze a sample of data collected from an overall population, and what's the best way to describe it?
So this is what we're going to talk about over the next half an hour, 40 minutes or so. What is data? How you can present your data based on sampling and variation within the data set that you may have collected. What are some basic concepts like correlation and causation, and how these can be illustrated, how these can be effectively presented on your figures and in your data tables. And we'll summarize at the end the learning outcomes, as I've discussed already, like what is it that you should be getting out of this presentation. And as I've mentioned, you'll have the opportunity to come back and ask us any questions as well at the end of this presentation. So what is data? Data is a collection of facts, things like values or measurements. And really, these are the raw materials, the building blocks of science. Contains information waiting to be released. And this information, this content within the kinds of sampling that you might do as part of your scientific work is done through interpretation, presentation, and statistical analyses. So in terms of the ways that you might present your data, you may present your data in terms of types, in terms of numbers, in terms of summary statistics, correlations and causations, and what will go onto those figures that you put into your articles, what will go into the summaries of your data, the summaries of your results that will sell the message of your study to those editors, to those reviewers, and to those readers.
Yeah, so I think this is the first time I'll get to say this, but
Yeah.
Every single data analysis in manuscript preparation is always an attempt to place a reader close to the actual data, because every study is so different, and you could have found pretty much anything. What you're describing here is an attempt to get to know the data in any given study. So I think this is why our authors need to have a good sense of how they might do this and what are the best ways to allow a reader to quickly and easily get a sense of their data. And I think this is why this slide is so important. It sets the stage for everything else we're going to talk about.
So here are some types of data. The kinds of data that you might collect in a particular study can basically be either qualitative or numeric. Qualitative or numeric, often referred to as quantitative in the second category. So things like qualitative data examples can be descriptive things, things that somebody might have told you in an interview, or your description of a way a building looks, for example. Whereas numeric or quantitative data, people much more readily associate with analysis, numerical analyses. These are nominal, categorical, ordinal, or number-based systems of data collection in a number of different forms.
That's right. And I think that system, the nominal, ordinal, interval, and ratio, that system is one of the most widely used way to describe data. Although many fields don't use it, it is a good way to think about the data, because if you could tell what type of data you have, if you could put it into one of those categories, you know a lot about what you've done. For example, if you know that your data are all nominal, they're all categorical data, that would lead to a very particular set of analyses. Whereas if your data are more ordinal or interval data, you could actually then use a different set of analyses. I don't think we're going to be talking much about qualitative data today. But the rest of our presentation, I think, don't you think, Gareth, will be about numeric or quantitative data?
Yeah, and something that people often do when they start a study, and it's worth thinking like before, because in my experience, students especially have gone ahead, rushed off and started to collect lots of data for a particular study without thinking in advance like what that means and what kind of analysis they might end up needing to do. So it's very well worth putting some thought into what the question is that you want to answer with your study, and therefore what kind of appropriate data would you need to collect to answer that particular question.
Absolutely.
So I just want to say as well, like just as we move through this, that when presenting data in scientific articles, when writing up your results, the results section of your academic papers, there's a number of general editorial rules that people tend not to follow, but probably should follow. And editors, professional editors, like the crew working for the Charlesworth group will always edit these kinds of things. So numbers up to including 9 in the text are always written out in full. So 1, 2, and so on. But larger numbers, generally 10, 11, and bigger, are written out in numerals. And we try to round up numbers up to 10,000. So keep it simple. And we'll talk about this when we come back to data presentation later. You probably want to give the reader something that they can grasp, like 6,200 rather than 6,248. You know, so these kinds of rules for editing and presenting data, numbers in the text of your academic papers are also quite important to keep in mind.
And you know what else, Gareth, is most journals have a variety, I should say, of reporting styles. And so the best way to see what a journal is looking for in terms of numerical reporting is to look at the instructions for authors for that journal, or even to examine a couple of articles published by that journal, because as the advice you have in this slide is very, very good. But many journals actually have specific ways they want numbers presented in different styles. So to get a sense of where you're going to submit your article will help you decide how to present that data as well.
Awesome. So in terms of sampling then, and I don't know if you want to talk about this slide, Tim, but either like sampling can either be non-probability based or probability based. So differences between the kinds of sampling approaches that you might use when doing an experiment, when collecting data.
Yeah, I think sampling, how the data are observed. So as I mentioned earlier, almost every study involves an investigator attempting to study a population of some kind, a population of people who suffer from a disease or a set of nervous system qualities. You have to come up with a way to observe the data, to create a sample. So how is that data observed? Well, that's an important consideration that is often ignored. Most research, I would argue, uses non-probability sampling. And that's when we don't really know how well the current sample fits the population from which it's intended to describe or be a representative of. And so I think if, for example, one type of this is convenience sampling. If you're doing an observational study and you only examine patients who present for treatment, well, that can be very interesting, and that can very much help you figure out a certain prevalence rate in those individuals. But it's not a guarantee that those individuals will be representative of the broader population that you're interested in. So there are very much limitations to the sampling design and what interpretations you can make. So just like knowing what type of data you have, knowing the sampling strategy you used in your study or will use can determine what type of interpretations you can make. And this is something worth knowing more about.
So we're looking at attributes that may vary in each experiment, in each sampling. Okay, so as we've talked about before, qualitative variables, which we won't discuss further, but aspects that can't be assigned numerical values. Quantitative variables, of course, things that you can actually measure, things that you can actually count or take out a tape measure and actually record, you know, the length of or the width of. These can be assigned a numerical value. So we're talking about kinds of data that could be discrete, where the values are distinct and separate, so they can be counted, in other words. Categorical data, where values can be sorted into different predefined, perhaps, categories, where you, the user, the experimenter, have placed like or defined a number of bins, let's say, that you might put your data samples into as you go through your experiment. So, you know, animals between a certain length or, you know, over a certain size, for example. Nominal data, where values can be assigned a code, where you're coding to approximate or to express a number, where the numbers become labels. Perhaps ordinal data, where you rank your values. You try to see, like, on a scale of 1 to 10 or on a scale of 1 to 100, what would be the rank, the position of that particular sampled element in the context of all of the experimental data. Or continuous data. I think perhaps one of the hardest kinds of data to treat statistically, right? Continuous data where values can take on any value within a finite or infinite interval.
I think that's true. If a data is continuous, viewed as a continuous scale, then you have to consider some other things, which we'll be talking about later today. One of the benefits, though, of using data types if you have available several different types of data, for example, if you're designing a study and you could reliably measure your data using continuous scale, like for example, let's say you're studying the a body mass index of a group of adults. That's a decent example of a continuously scaled data. You could view that as it is, as BMI, or you could create a categories, like for example, BMI above or below 25, creating a category. Well, the benefits of having continuous data is that they contain more information. And data that contains more information will be more powerful in terms of how you analyze them with statistical approaches. So oftentimes, if you are able to reliably measure something with a data that has more information, you'll need smaller sample sizes. So when I help my investigators that I work with design studies, I say, let's try to find data that have the most amount of information in them and use that representation of the data. Does that make sense, Gareth?
Yeah, I think so. I mean, in my experience, it's very difficult sometimes to design appropriately the experiment beforehand
Right.
to make sure that the kind of data that you're collecting is going to be meaningful. And as we mentioned already, like so many examples that I've seen of people just collecting as much data as possible, assembling as much as possible in terms of their sample, and then thinking, what am I going to do with all this data? How am I going to analyze this, now that I've collected it? So these steps and how to think about designing and what kind of, just basically, like, what are you trying to test? What kinds of data are you trying to collect? To test those questions are well worth putting the time in to think about before you get started. So how can you summarize data once you've collected it? And one good way to do this is in the form of a table. So summary tables are very often seen in analytical papers, presenting data maybe in the body of the paper itself, or perhaps in the appendices on the online sections of your study. And just some examples here of what like a frequency table, a summary data table might look like as somebody is presenting the kinds of data that they may have collected in this particular study, for example.
Yeah, I think the use of tables is an art form. I really believe that the difference between a good paper and a great paper is the use of tables and figures. And I know we'll be talking more about that later. But oftentimes, authors don't present their data in a very clear way in tables. And that creates a problem for reviewers and editors because they can't evaluate what was done here and what the authors found. So I think taking your
Yeah.
time and really thinking about data layout on tables is crucial.
We'll talk about it in a little bit as we move through this presentation, but be consistent. Like as an editor working for many years, editing lots of papers, like I like tables in papers because they get counted as part of the word count of the study. And so an editor will see a table and think, wow, great, like that's, 1,000 words that I don't have to really edit. But that's often a mistake because some of the hardest work for editors working through papers before they go to journals for review is to make sure the tables are consistent. So you as an author can really help your reviewers and your editors by picking a style and then sticking to that style. Not just in the one paper, you may choose a style or a way of summarizing data using a piece of software like Excel that you can then use in all your papers, but keep it consistent, I think is good advice when presenting data. So we'll talk about summary statistics and how you can put together a series of summary statistics in your studies to summarize sets of observations to communicate the largest amount of information as simply as possible. So some of the simplest kinds of data-based statistics that you'll see in numerical studies are just things like measures of location. What's the average, the mean, the median, the mode? What's a measure of spread? Something like the standard deviation, like how spread out is your data versus the mean, for example. What is the shape of the data distribution? How skewed is it? How disparate is it? And you can identify this kind of thing from your data very easily by simply plotting a histogram. So basic summary statistics that people will often report in studies before they go on and talk about like an analytical example, like just what does the data show? After summarizing it in a table, You then want to tell the reader, tell the reviewer, what does the data show in terms of a summary series of statistics?
Boy, that's a great introduction, Gareth. I'm going to say something that I think is important. It is, if our listeners today take one thing away from this presentation, it should be that descriptive statistics, data used to describe their samples, are the most important thing they can do in data presentation. Describing what you observed in a way that is easily accessible and that can actually describe the sample in a way that allows a reader to understand what was found is crucial to understanding what's happening. Most times, young authors ignore this element or don't present enough descriptive statistics. They go right to statistical testing or p-values or more of the sophisticated analyses, and they ignore the importance of descriptive statistics. So I think the single thing you can do to make your papers more easily read and more impactful is to present a lot of descriptive statistics. Every time you're trying to make an argument about something, provide some descriptive statistics about it.
Just basic stuff, as we talk about on this slide. How many papers have you read where you just don't understand what's going on? You think, this is just so heavy with the analysis, I've lost track of what the paper's actually trying to tell me. And often I get that feeling with many papers that I read and review. You're like, this is so analytical, I've lost track of what the main message of the study is. And as
That's
Tim
exactly
says,
right.
that are the suggestions for software packages that people might wish to use.
Great.
We'll talk about figures as we move on, and thanks, Tim. That was really informative and really helpful to go through a lot of that basic context for the kinds of techniques that people can use. Once you've collected data and thought about what it means, summarized it, let's say tabulated it, talked about what the basic summary statistics are within your data, and then perhaps performed some more complicated analyses to get that message across to people in the paper. Now, what about putting figures together? And a key part of statistics and data presentation is the development of figures. You always must make sure that your fonts and sizes are large enough for people to read. I've seen so many figures in scientific articles that you can't easily read. Make sure you reference all the figures in the text of your article. I mean, there's no point including figures in a paper if you're not going to talk about them in the text. And very important thing to remember when putting together figures for scientific papers is to give them self-contained captions, because very often people take just the figure off of the PDF or off of the journal website, and they need to have enough information in the caption to interpret the figure without having to read any more information from the rest of the paper itself. So basic ideas about being effective in your figure presentation. There's a fantastic paper that was published in 2014 that's referenced here on this slide. 10 rules for making better figures, from Public Library of Science Computational Biology. And I recommend that everybody goes and gets a copy of this paper. The slides that you're going to see now come directly from this paper, and there's 10 basic rules to make effective figures in terms of your data presentation. Know your audience is the first one, and we've talked about this already. A paper that you might write for Nature or Science, very different to the kind of paper that you might write for a specific journal in your field. How to present your data to the appropriate audience is very important when putting your figures together. Make sure you know what the message is in the figure. And this goes back to what we talked about at the beginning of this webinar. If you know this before you start to write, start to do the analysis, then doing things like presenting tables, putting together figures can really be much, much easier, because you know what you want people to get out of the paper. You know what the message is. Construct your analysis, construct your figures around that core message. And that's the second rule for making great figures. Make sure you adapt yourself to the medium, and make sure you eliminate noise from your figures, as shown here quite nicely on this two examples of the same data. But do we really need to show all of the particle tracks in this particular figure to get the message across? Of course not. You can summarize, you can adapt to the kind of, publication medium that you're going to be using in your particular presentation. Captions are not optional, is rule number 4. And make sure that you pick a software package that you're comfortable with using, that you know how to use, and that will produce reproducible looking figures. So the software that you use has to be something that you're good at using, that you're comfortable with using, and that makes your data say what you want it to say. So many people in their papers use different software packages for different figures in different places, and they look different, even if the graph is, basically the same, or the data being plotted is basically the same. Software, it's very important. And the use of color in figures in articles is also hugely important, as shown here. And there's a massive body of literature that's been written about the most effective way to use colors to draw people's attention to your figures and get your message across most appropriately. It's very field dependent, depends on what you're trying to present in your papers, but think about the most appropriate contrasts to make your figures look as effective and summarize your data as well as possible. And don't mislead people. Often you see this in papers looking at data analysis, and you can see this here. Depending on the axis of the graph on the right-hand side of this slide, the trend in the data looks bigger or smaller to the reader. The same with using relative area of a disk to summarize data as well. People often use these kinds of techniques to get people thinking, oh, well, I can see there's definitely a trend in these data. And that's important to keep in mind, to think about when you put your figures together. You don't want readers, reviewers in particular, to criticize this aspect of your study. He just, shifted the axes on a graph, for example, to make the trends look bigger than they actually were. I've talked about this already.
That's a great point. Sorry to interrupt you. That's a great point,
No.
because I think authors can get into big trouble with journal editors and reviewers if they manipulate the axis to make something look bigger than it is, or to obscure the findings. I've encountered several different interactions with authors where they've actually been banned from journals because
Really? Wow.
it appeared as if they were openly trying to manipulate the findings, etc. And I think you have to be careful how you do that, and there are rules for doing so. All our authors should be careful about it.
Yeah, and we've got a lot of material. The Charlesworth group, we have a lot of educational material that you can look at. You can get in touch with us to talk about afterwards about how to do this, how to put figures together in the most effective way. So don't mislead people, and avoid junk on your graphs as well. And this is another example of a completely confusing time series graph on the left-hand side that gets the message across, but it's really hard to interpret, isn't it? Would have been much better if the authors had scrapped the color in this case and just presented their time series separately, so people can see much more easily what's happening in this figure. So put a lot of thought into your data presentation is the message of this section of the presentation. And often really, like, the message that you're getting across to people is more important than how good your figures may look, as shown in this nice example here. I mean, it's not how good your figures look, how beautiful your artwork is in your scientific article, in your academic article. It's whether or not you actually show what your data shows, and whether or not you can support what your data shows with analysis and in the figure itself at the end of the day. So that's rule 9. Think about messaging rather than beauty in these slides. And rule 10, again, as I've talked about already, get a tool, get a software package that you know how to use, and then use it consistently, even if it's something simple. I always use tools like Acrobat, Illustrator, Adobe products when putting together figures in my own work, and that's just because I like to use those kinds of software packages, and I know how to use those kinds of software packages. It depends on you, it depends on your field, but Be consistent and make figures, make tables that are consistent across perhaps all of your academic work in the future. Develop a style for your illustrations. Tables are important, and I just quickly will talk about tables because I'm aware that we're running out of time in this webinar today. But tables, as Tim mentioned earlier, are an art and a way of presenting data that makes your data much easier to understand. A standard way to present and summarize your findings. Readers don't have to struggle to find information in your tables. And if you make them look good and you make them look consistent, you make yourself look much more professional as well. And it's a quick and easy way to support the conclusions of your study, as we've talked about already at the start of this presentation. So here's an example of a misleading table on the left-hand side of this slide. The population of Wales, which is a region in the United Kingdom. The trend here is not important. The number of values are quite small, and you must get the message across in the way that you present the data. So putting all of it into the table can be unnecessary and can be confusing in some cases, as shown on this example here. So just a few quick style rules for you that you can look at in your own time when you come and review this presentation, when you look at the video, things you should do when you style your tables and when you summarize your tables. Don't produce tables that look like this. I've seen many, many examples of these in the papers that I've edited over the years. Different fonts, different styles, different formatting for the data. You want something that's consistent, that's targeted to your audience, and that gets across the message that you want to present in your article. And create, please, a usable template for your future work so that, as we've mentioned, you develop a particular style in your articles that people can tie back to you and that is effective and simple in your kinds of communication. Histograms are one way to graphically represent the contents of a frequency table. I don't know if you want to say a few things about histograms quickly, Tim.
Yes, as we mentioned earlier, histograms are one of the best ways to examine your sample to see what type of descriptive statistics that should be applied and perhaps to inform the type of analysis you might want to conduct. But as Gary's saying, now when you're going to present the data, oftentimes a histogram can be the most effective way to describe either the frequency counts observed in certain categories or differences between two different groups in terms of the distribution of scores. So a histogram should be a very easy way to present a message that might be appropriate for your paper. I use them a lot.
Yeah, to deal with large data sets, to present outliers and gaps, and to quickly and simply illustrate to your readers what the distribution of your data looks like. Very, very effective. Pie charts too can be useful to represent categorical data, to summarize percentages, because of course, as you know, the areas in these charts are going to be proportional to the value of each category, but they don't have a time series on them. So these kinds of charts should not be used to present historical data over time, for example.
Gareth, a quick comment for you. I have one too.
Yeah,
I think a lot of authors present 3D plots when 3D may not be necessary. So in this case, this is an attractive looking pie chart for sure. But I think adding 3D features to plots can oftentimes be annoying to reviewers and editors. For example, one of the journals that I work, we try to outlaw 3D plots when they're not required.
That's a good point. Very good point. Try to keep it simple. It's a key
keep it simple.
take-home message from all of this. So what do you prefer? Which kinds of data summaries are more attractive to you? Have a look at this image in your own time when you look at this presentation again. Which is the easiest way of summarizing the distribution of a healthy diet? What is better? What do you find more attractive in terms of data presentation? I know which one I find easiest to interpret, but I'll be interested, maybe we can talk about this offline later, but which of these plots is better in terms of data presentation? Lots of tools Tim can talk more about this, of course, as well. Plenty of tools are available to plot and do statistical analyses. There's a couple that you may have heard of, MATLAB and Excel are the two that I use most often. I actually recommend R. I don't know about you, Tim, when it comes to doing these kinds
I
of
use
analysis.
R and R Studio for all of my analyses, and I think it's certainly my favorite statistical software package, but it does have a lot of learning curve to it. It's difficult to learn.
Yeah, open source. But yeah, I mean, like in terms of different packages that you may wish to have a look at, get back to us and think about things that you may use when analyzing your data. Here's a few suggestions for further reading that we put together. Great TED Talk, the best stats you've ever seen, and also this Rougier 2014 paper from PLOS Computational Biology that takes you through what I call the 10 rules of the pirate code for putting together effective data presentation packages in your own articles. Thank you so much for listening, and thank you, Tim, for participating today. It's been great to have you on board with this statistics and data presentation webinar. Thanks a lot for your time.
Thank you, Gareth. I had a lot of fun.
This is our website. Please have a look at some of the other material that we have available that may help you as you develop your career as a young researcher. Every Tuesday now, at the end of each month, we have a webinar brought to you by Charlesworth Knowledge. Please check out the rest of our series. Why not spend the last Tuesday of each month with the Charlesworth group learning something important, potentially informative to advance your career. I mentioned a special gift at the beginning of this presentation. Here's a 10% discount code that you can enter for the next month into our website and get 10% off all of our editing services. So language polishing, pre-peer review, author services, including, we're very excited to announce, a statistics review piece of software that Tim and his colleagues have developed that will tell you if your paper is appropriate in terms of the statistics that are included. Would that be a fair assessment of what your stats reviewer software does?
I think so, yeah. We at Stat Reviewer are excited to be partnering with Charlesworth to help your authors pre-review their manuscripts for quality of statistical and methodological reporting. We think that the reports can help them improve their manuscripts and make their work more likely to be published.
So you can get 10% off all of these services if you plug this discount code into our website. And I will stick around, Tim will stick around now for the next few minutes. I know this webinar's gone on for a little bit longer than expected, but thanks for sticking around. I see that many of you have stuck around, so it must have worked. Thanks very much for your time. Please put some questions into the chat box. I'll keep talking. We'll just have a few minutes of silence while I collate my notes and so on, and then we'll come back and we'll answer some of the questions that you guys have. Thanks again, Tim, for participating, and thanks to everybody for listening. Please come back to us with any questions that you may have.